As we age, our cells and tissues lose their regenerative capacity, making our bodies less efficient at repairing themselves. Rejuvenating medicine focuses on reversing aging and its consequences by trying to restore the regenerative capacity of stem cells. To preserve their regenerative capacity, stem cells must harmonize between self-renewal and differentiation. This is achieved by balancing symmetric and asymmetric cell divisions, and it is linked to the capacity of establishing cell polarity. One of the key factors involved in this process is CDC42, which is overactivated in aged mice and humans, and is linked to the aging of stem cells, premature aging, and a shorter than average lifespan. To target CDC42 overactivation and enhance the regenerative capacity of aged mice, scientists treated old mice with CASIN, a molecule that specifically reduces CDC42 cell activity. This treatment managed to extend health span and lifespan of old mice. CASIN also caused structural and epigenetic clock changes in aged mice that suggested that cells and tissues in the treated animals were reset to a more youthful state. Moreover, CASIN treatment also helped restoring the animal's aged skeletal muscles and increased the animal's average strength. To investigate the dependency of this effect on stem cells, the research team conducted a transplantation experiment. They transplanted blood stem cells from aged mice treated with CASIN into untreated aged mice. The result was that aged mice that received these rejuvenated stem cells lived significantly healthier and longer compared to the non-treated ones. Therefore, rejuvenating endogenous stem cells with treatments such as CASIN might soon become an effective strategy for delaying aging and preventing age-associated diseases in humans.